I'm Eric Marsh. I am a child neurologist and epileptologist and researcher at CHOP. And I've been a member of the advisory board for the LGS Foundation for a number of years. So it's great to see this come to fruition and watch the LGS Foundation really grow and expand and consider new ways to do research about LGS. And that's one of the main tasks of this, is to figure out the research priorities. And so, we're taking stock of LGS-related research. And as you heard this morning, as Tracy just said, we know an awful lot, and though we still have more to learn about what is LGS and how to treat LGS, but there's still an awful lot to learn. And one of the key features, as described this morning, right, is there are a lot of different etiologies of LGS. But the shared features are those slow spike wave and GPFA and standard seizure types, which raises the question is, what is at the heart of that shared component? And is that really kind of what we should be researching as LGS researchers? Should we be focusing on what are those network mechanisms that drive slow spike wave and GPFA and these seizure types? And what are the underlying clinical features that get us there? And can we find shared clinical features amongst all these different disorders to get a better understanding of really what is the core of LGS, and not the individual conditions, but really what is at the core? And so this afternoon, we're going to start to try to ask some of those questions by researchers who've been doing this. And we really want to get into understanding kind of how we can use clinical data, both chart, EMR-based data, and clinical data from EEG and MRI to understand the network and these core features of LGS. And then we'll end this session with thinking about, once we know that, how would we design a clinical trial, or how would we move LGS research forward? And so I'm going to now hand it back to Brenda, who's going to give an introduction of all the speakers so that we can just go from kind of one speaker to the next. Brenda?
Thanks. So I think that we did, Eric and I spent some time with Tracy and the group on trying to find a group of speakers that all are addressing the question of what is LGS, but from different angles. So it's a synergistic panel, I believe, that have different research focuses, but they're all, I think, going to bring a lot to the table as far as what is LGS and how do we study it. So the first person that's going to speak is Dr. Jurriaan Peters, who is a good friend of mine. He's an associate professor, I believe. I have assistant.
Not yet.
Oh.
A few more months.
Okay.
I just were promoted, Jurriaan.
We're all getting new titles. This is very exciting. Okay. And his expertise is in the EEG changes that accompany the development of LGS, particularly with the disordered tuberous sclerosis complex. So he'll be speaking on that. And then Dr. Jennifer Galinas is a MD PhD, assistant professor of neurology at Columbia. She is a pediatric neurologist, and her interests are really focused on understanding how the epileptic network disrupts normal development. And she's used intracranial EEG and other sort of electrophysiologic biomarkers to study children with epilepsy. Next, we have John Archer, who is a senior lecturer at the University of Melbourne, and we owe a special thank you to him because it's very middle of the night in Melbourne. And he's going to be joining us primarily, I think, talking about different ways that they've studied the epileptic network in LGS. And I owe a big thank you to him. He, several years ago, convinced me to try to do a DBS in the central median nucleus because of the study they had ongoing. And he's been very generous since then. And I found it's been helpful to many of my patients. So I appreciate that. And then Dr. Greenspan, Zachary Greenspan, is an associate professor of pediatrics and population health sciences at Cornell. And he is a pediatric neurologist and epileptologist, and he's very interested in how to use the medical record system to study more rare epilepsy syndromes. But he's also going to, I think, focus on identification of at-risk patients, and how, you know, we should be thinking about them as trial subjects. And then finally, we have Dr. Dennis Delugos, who is a professor of neurology and pediatrics and taught me most of what I know about EEG. And he's very interested in the question of how to design clinical trials that are more efficient and more successful. So he'll be our final speaker, and then we'll go ahead back to kind of a discussion section.